Hello, learners. Every single cell in your body carries the exact same DNA, the same genome copied identically from a neuron in your brain to a muscle fiber in your arm to a red blood cell racing through your veins. So here's the question that should stop you in your tracks. If they all have same instructions, why does one become a brain cell and another becomes your heart? Why does one carry oxygen while another lets you lift a weight? The answer isn't in having different DNA, it's in how the DNA gets used. And that story begins with something called a gene. Welcome back to Gene Up Informatics. My name is Komal. Let's get into it. Think of your DNA as Netflix. Just like Netflix holds thousands of movies and shows, your DNA holds thousands of genes. And just like every movie on Netflix has its own unique story, every gene carries its own unique instructions. So, what exactly is a gene? It's a specific segment of DNA that holds the instructions of build something, usually a protein, which then goes to perform a specific job in your body. One DNA, thousands of genes, thousands of functions. One more time, in detail, let's see what is a gene. A gene is a functional unit of heredity. It's a specific segment of DNA that carries the instructions to build something useful. Sometimes that's a protein, like an enzyme or a hormone. Sometimes it's a functional RNA like tRNA or miRNA. And sometimes it's other RNA molecules that help regulate how your genes behave. But here's what makes genes really powerful. They don't just build things inside you. They get passed down. From parents to children, genes carry the traits that make you, you. Now let's break open a gene and look at its anatomy. Every gene has multiple functional regions and each one has a specific job. First comes the promoter. This is where RNA polymerase binds and transcription begins. Inside the gene itself, you have exons and introns. Exons are the coding parts. They stay in the final RNA and usually carry the protein binding instructions. Introns sit in between and they actually get removed later during a process called splicing. And finally, there's the terminator, which simply signals stop, transcription ends here. So here's the full journey. DNA gets transcribed into pre-mRNA, which contains both exons and introns. Splicing removes the introns, leaving mature mRNA with just the exons. The mRNA is then translated into a protein. Same gene, but single region controls when, where, and how that protein finally gets made. Now here's a question people often assume the answer to. Are all genes protein coding? The answer is no. Protein coding genes go through transcription and translation to produce proteins like hemoglobin, insulin, or keratin, the ones doing structural and enzymatic work in your body. But there's a whole other category, non-protein coding genes. These produce functional RNAs instead. rRNA forms the core of ribosomes, tRNA brings amino acid during translation, miRNA regulates gene expression, and link RNA plays a role in various regulatory functions. These RNAs never become proteins, but they still run the show behind the scenes. And here's the interesting part. Out of all your genes, only about 20,000 actually code for proteins. 1,000 more are busy producing these functional RNAs instead. Here's something that might surprise you. One gene doesn't always mean one protein. This happens through a process called alternative splicing. In normal splicing, all exons are simply joined together to form the final mRNA. But cells have other options too. Sometimes an intron gets retained or an exon gets skipped entirely. Other times, the cell uses alternative splice sites, changing exactly where the cut happens. So the same gene can be spliced in different ways, creating different combinations of exons. Different exon combinations means different mRNA molecules, and different mRNA molecule mean different proteins, all form a single gene. This is exactly why humans have only around 20,000 protein coding genes but end up producing hundreds of thousands of distinct proteins. Alternative splicing is one of the biggest reasons your body can build so much diversity from so little code. So what happens when a gene changes? This is called a mutation. Simply a change in the DNA sequence. Mutations can happen in a few different ways. A substitution swaps one nucleotide for another. An insertion adds an extra nucleotide into the sequence. And a deletion removes one entirely. Now here's the important part. Not all mutations are bad. Sometimes they have no effect at all. Sometimes they can even improve adaptation. But other times they disrupt protein function or cause disease. Conditions like sickle cell disease, cystic fibrosis, and BRCA1 mutations like linked to cancer risk all trace back to changes like these. So remember, mutations are simply natural changes in DNA. They can be neutral, beneficial, or harmful, and together they have been quietly shaping evolution and health for millions of years. So why don't all cells use every gene? Even though every cell contains nearly the same DNA, different cells switch on different sets of genes. This process is called gene expression. A neuron activates neurotransmitter related genes. A muscle cell switches on contractile protein genes. A liver cell relies on metabolic enzyme genes. And an immune cell activates immune response genes. So DNA never changes. What changes is which gene get turned on. And that's really the answer to the question we started with. Cell identity doesn't come from having different DNA. It comes from which genes are expressed. Genes are the central focus of genomic research, and here's why scientists spend so much time studying them. They investigate genes to discover disease-causing mutations, comparing genomes across species to understand similarities in DNA, predicting protein functions from gene sequences, and studying evolution by tracing the history of genes over time. They also use genes to develop precision medicine, tailoring treatments based on person's individual genetic makeup, and to design gene therapies that target genetic disorders directly. So at the end of the day, genes are so much more than just DNA. They are the key to understanding life itself and to improving human health. Every sequencing project, no matter what it's studying, eventually asks the same set of questions. It starts with sequencing the DNA, then assembling the data into a genome. From there, bioinformaticians move into gene prediction, figuring out where the genes actually are, followed by gene annotation, labeling what each gene does. Next comes variant identification, spotting differences or mutations, then functional analysis to understand what those variants actually mean. And finally, biological interpretation, connecting all of it back to real biology. Whether you're analyzing whole genomes, RNA-seq data, metagenomes, or cancer genomes, it always comes back to the same goal. Identify the genes, understand their function, and figure out how changes in them shape biology. Genes really are the foundation of nearly every bioinformatics workflow. Let's quickly recap everything we have covered about genes. A gene is the functional unit of heredity. It contains instructions for producing proteins and functional RNAs. Structurally, a gene consists of both regulatory and coding regions, and not all genes actually produce proteins. Some produce functional RNAs instead. One gene can even generate multiple proteins through alternative splicing. Mutations can alter gene function and contribute to disease. And finally, gene expression is what determines the identity and function of each cell. Same DNA, but different genes switched on. And that's gene from structure to function to expression. So what next? In the next episode, we are going from gene to protein, exploring central dogma of molecular biology. DNA contains the gene, the instructions. Through transcription, the gene gets copied to mRNA, and through translation, that mRNA gets built into a protein. We'll be answering questions like, how does a gene actually become a protein? What is transcription and what is translation? Why is mRNA so important? And how does all of this connect to real research like RNA sequencing and transcriptomics? So get ready, because next episode we are diving deeper, connecting everything we have learned so far to real genomic projects. Follow us on Instagram for bioinformatics tips, learning resources, and updates about upcoming videos. Stay curious, keep learning, and keep growing. Thank you for watching, and I'll see you in the next video.